Not long into your study of biology, you've probably already learned about the 4 macromolecules central to living things: carbohydrates, lipids, nucleic acids, and proteins. Protein is hugely important to living organisms by providing structural support, system regulation, immune defense, cell transport, motion, and metabolism. So if protein is so important, it must be created constantly to keep up the supply. Yes, proteins are made, or synthesized, in all cells in the body all the time in a process called protein synthesis. Protein synthesis can be understood in 2 major steps: transcription and translation. It might help you to remember which step comes first in the process by noticing that there is a C in transcription and an L in translation. C comes before L in the alphabet, just like transcription comes before translation in protein synthesis. Now that we've covered the basics, let's get into the details. Transcription, the first part of protein synthesis, takes place in the cell nucleus and involves 2 major players, DNA and mRNA, or messenger RNA. Think about the job of a messenger, to carry information from one place to another. Messenger RNA, mRNA, does just this. The mRNA leaves the nucleus with its information and attaches to a ribosome in the cell. Remember that ribosomes are actually made up of 2 subunits, one large and one small, composed of rRNA, or ribosomal RNA. Once the mRNA has arrived at the rRNA, the ribosome, the message will be used to create protein. This is where translation begins. Think about the job of a translator. He takes information from one form and presents it in a way that someone else can understand. This second part of the process takes place on ribosomes in the cell and involves mRNA, tRNA, and rRNA. tRNA, another type of RNA found in the cell, is now part of the process. tRNA, or transfer RNA, carries amino acids, the building blocks of protein. The tRNA is looking for mRNA containing information it matches with. When they match, the tRNA drops off its amino acids, and the amino acid bonds with the mRNA to make protein chains. This process, repeated over and over again, results in long strings of protein, each specifically created to serve an important and distinct purpose.